Adaptive immunity comes into the picture when the innate system is not sufficient against an established infection. In contrast to innate immunity, which is non-specific, adaptive immunity generates a pathogen-specific immunologic response to eliminate the target microorganism. The cells involved in adaptive immunity can also develop an immunologic memory so that, in future, if the same pathogen attacks the body, the reaction will be faster, stronger, and more effective in neutralizing and clearing the pathogen. This is the principle behind vaccination. A vaccine usually contains an inactive form of the pathogen which generates an immune response and develops immunologic memory without harming the body. In future, if the pathogenic form of vaccine infects us, our immunologic memory reacts faster and protects us from the infection. The adaptive immune system mainly has two types of lymphocytes. One is B lymphocytes, that is B cells. The other one is T lymphocytes, that is T cells. B lymphocytes are called B cells as they mature in the bone marrow. So we are all aware of antibodies, and these antibodies come into the picture now. When a B cell leaves the bone marrow, they express these antibodies, which are nothing but antigen binding receptors. That is, a B cell binds to an antigen through these antibodies. A B cell that has not encountered any antigen is known as a naive B cell. Once it encounters a specific antigen, it activates B cell to multiply rapidly, forming two kinds of progeny, that is daughter cells. One is memory B cell that has a long lifespan and express the same antibody as its parent B cell. The other progeny is plasma cell that secretes enormous amounts of antibodies. They may have little or no bound antibodies on their membrane as they secrete everything. They have a short lifespan. T lymphocytes mature in thymus gland and therefore the name T cells. During maturation, they express T cell receptors. Unlike B cell receptors, that is antibodies, which bind to antigens alone, T cell receptors only bind to antigens that are already bound to cell membrane proteins called major histocompatibility complex, that is MHC molecules. These MHC molecules take part in a process called antigen presentation. There are mainly two types of MHC molecules. Class 1 MHC molecules, which are expressed by nearly all nucleated cells, that is our body cells, which are generally considered as self cells of the body. If these self cells display foreign antigens that are complex with a class 1 MHC molecule, they are called altered self cells. In general, normal self cells have become abnormal, maybe due to infection or tumor or cancer. Class 2 MHC molecules are expressed by only antigen presenting cells such as macrophages, B cells, and dendritic cells. So, T cells have the property to recognize only particular antigens that are associated with MHC molecules. It means it can discriminate which is normal body cell and which is abnormal. This property is called self to non-self discrimination, and it is a unique property of our adaptive immune system. Similar to B cells, when a naive T cell encounters an antigen combined with an MHC molecule on a cell, the T cell proliferates and differentiates into memory T cells and various effector T cells. T cells have subpopulations called T helper cells, which have CD4 glycoproteins on their surface, and T cytotoxic cells, which have CD8 glycoproteins on their surface. In adaptive immunity, the clearance of a pathogen occurs by humoral response or cell-mediated response. Humoral response occurs when a B cell interacts with an antigen. As I told earlier, the interaction of B cell with the antigen leads to proliferation and differentiation of the B cell to antibody-secreting plasma cells. When an antigen is coated with antibody, it can be eliminated in several ways. Antibody can cross-link several antigens, forming clusters that are more readily ingested by phagocytic cells. Pathogens generally bind to host cells through the receptors. These antibodies coat around a pathogen and block these receptors so that they cannot bind to host cells, and therefore infection is inhibited. This is called antibody neutralization. Cell-mediated response occurs through T cells. Both Th cells and Tc cells take part in cell-mediated response. Th cells are activated when the receptors recognize antigens bound to MHC class II molecules. Activated Th cells secrete cytokines that in turn activate B cells, Tc cells and macrophages, which eliminate the pathogen by the respective methods of pathogen elimination. The other way is T cell activation. Cells that are infected by pathogens such as virus-infected cells degrade virus particles inside, and degraded viral antigens along with class I MHC molecule is expressed on their membrane. Tc cells get activated when they interact with these complexes. Activated Tc cells proliferate and differentiate into cytotoxic T lymphocytes. These cytotoxic T lymphocytes exhibit cytotoxic activity, that is, they kill the virus-infected cells and prevent the spread of infection. Gradually, the established infection is reduced and the pathogen is completely eliminated from the body.